In 1875, a book published by Charles Darwin sent shockwaves through the scientific community. Some believed that the theories in his insectivorous plants were utterly absurd, nothing short of a sacrilege to science. Darwin's discovery stemmed from his study of a specific plant. He noticed that the sticky tentacles of the sundew, which grew in southern England, were often covered with the carcasses or remains of up to a dozen insects. Insects being killed by plants is not entirely uncommon. In fact, many plants cause insect fatalities, such as those trapped by the protective mucilage of the horse chestnut, which often meet an accidental death. But Darwin had a sneaking suspicion that the fatalities on the sundews were fundamentally different from those ordinary accidents. He suspected these insects were deliberately hunted by the sundew. To prove his hypothesis, Darwin began collecting specimens for experiments, attempting to demonstrate whether plants could actually trap, digest, and break down insects for their own use. Today, no one doubts the existence of carnivorous plants. There are over 600 known species of carnivorous plants, each employing distinct hunting skills. The sundew that Darwin studied relies on sticky secretions to ensnare its prey. Pitcher plants create inescapable pitfalls, trapping prey inside their jugs to be slowly broken down and consumed. Carnivorous plants have even evolved aquatic versions. This extreme member of the sundew family completely abandons its roots upon reaching maturity, relying on a trap that snaps shut in just tens of milliseconds, claiming the record for the fastest movement in the plant kingdom. Yet, when it comes to the most iconic carnivorous plant, nothing matches the visual impact of this terrifying biological bear trap. Take the Venus flytrap, for instance. Unsuspecting prey is lured by sweet nectar, landing right inside the massive trap laid by the plant. Soon, they are locked in a cage, never to escape past the fangs of this ravenous plant, only to be slowly digested over the course of the following week. What drove these plants to abandon self-sufficiency and instead move in the opposite direction, evolving into predators? And how did they become such deadly hunters step by step? In the wild, the Venus flytrap is found only in specific parts of the coastal plains of North and South Carolina. Their habitat, stretching across 200 kilometers, consists of broadleaf forests cleared by wildfires. Stripped of the dense canopy above, these low-growing plants finally get their share of sunlight. The wetlands serve as their killing fields. The plant folds its leaves down the center to form two lobes, striking a pose laced with sweet nectar, and then simply waits in silence. As small creatures like spiders, flies, and beetles crawl across the open trap, a countdown to the kill begins the moment the sensitive trigger hairs on the trap are tripped. However, if only a single hair is touched, the trap will not spring. Instead, if a second hair is tripped within the next 30 seconds, the cage snaps shut in the blink of an eye. This movement of the lobes is controlled by electrical signals. When a hair is touched and bent, the lever action stretches the membranes of the cells at its base. This opens a channel for positively charged calcium ions, which rush out to generate an action potential. This electrical signal travels from the hair throughout the entire lobe, causing the two halves to rapidly flip from being concave to convex, successfully springing the trap. The reason it requires two trigger hairs to be tripped within 30 seconds is to prevent false alarms caused by debris like raindrops, which would waste precious energy. But how does a Venus flytrap know that 30 seconds have ticked away? How do plants keep time? To test the calcium hypothesis, researchers genetically modified a Venus flytrap to emit a green fluorescent glow when calcium concentrations increased. The moment the flytrap detected the first mechanical stimulus, the calcium concentration inside the cells surged, immediately emitting a green glow. As time passed, the green fluorescence faded, indicating a gradual drop in calcium levels. If the sensor detects a second mechanical stimulus, the calcium concentration spikes even higher. The trap is triggered only when the calcium concentration reaches a specific threshold, which can only be achieved by two stimuli within 30 seconds. Yet, having an ingenious trapping mechanism isn't enough. The Venus flytrap also needs to know whether it has caught something useless. If you happen to own a Venus flytrap, you can replicate the experiment Darwin performed back in the day. If you trigger the sensors twice in a row with a piece of cheese, the trap will indeed snap shut. However, just a few hours later, it will reopen. This is because the trap will only stay closed and complete the digestion process if a living prey continuously triggers the sensors. This ensures the plant doesn't waste energy in vain. Once the prey's identity is verified, digestive fluids flood the sealed trap. The prey will struggle in agony for about a week. In a process akin to digestion in an animal's stomach, digestive enzymes break down the prey within the low pH environment of the fluids until it is fully liquefied. Finally, the inner walls of the lobes absorb the nutrients. Although the Venus flytrap absorbs nutrients from its prey much like an animal, the true purpose behind its hunting is not energy, it is nitrogen. Without nitrogen, chlorophyll cannot be synthesized, meaning the plant cannot photosynthesize. Nitrogen is also an essential element for protein synthesis. The soil in southern North Carolina, where the flytrap thrives, is highly acidic and severely nitrogen deficient. Between 50% and 75% of the nitrogen the Venus flytrap requires is obtained from hunting insects. This phenomenon is not an isolated case. Their evolution from purely photosynthetic plants to carnivores was driven by the need to combat nutrient-poor soil. The waterwheel plant, the Venus flytrap, and the sundew share a common ancestor. Roughly 70 million years ago, their common ancestor underwent a genome duplication, creating an extra copy of its DNA. While one set of DNA began to mutate, seeking out new evolutionary paths, the other set retained the original DNA to maintain vital functions in case lethal mutations occurred. This strategy clearly paid off. For instance, normal plants use digestive enzymes to break down the chitin in fungal cell walls to fend off fungal attacks. Carnivorous plants, however, repurposed this ability to digest insect exoskeletons, which are also made of chitin. Ordinary plants rely on roots to absorb nutrients from the soil and produce nectar in flowers to attract pollinators. These same genes were repurposed by carnivorous plants for their traps, making the traps their primary nutrient-gathering organs. Following these mutations, different carnivorous plants occupied various ecological niches in their respective environments. Once the new survival strategy proved successful, traditional roots and leaves were no longer essential. The waterwheel plant completely lost its roots along with many related genes, becoming one of the plants with the smallest genomes. Interestingly, not all carnivorous plants share a single common ancestor. Existing carnivorous plants stem from at least 6 completely independent evolutionary lines. Although their predatory methods are remarkably similar, they are on different branches of the evolutionary tree, serving as textbook examples of convergent evolution across different historical eras. The most recent evolution of carnivory occurred in a member of the bromeliad family, only a few million years ago. The most fascinating takeaway from the study of carnivorous plants is that the mechanisms used to evolve carnivory are the very same pre-existing pathways that plants use to control their roots and flowers. In other words, if kept in a nitrogen-poor environment for a sufficient period of time, today's photosynthetic plants could, and likely would, evolve new predatory traits. Plants could very well become far more terrifying than the ones we know today.